perhaps being last in the session, I'll rush through. We should be taking you through a saga of research work that has been carried out in the lab and with my partners in Australia and Canada. And should be essentially speaking on how we deploy the genes that are there lying in the wall through genomic assisted feeding into modern chickpea varieties that have heralded India as the global leader in genomic assisted feeding. Next, please. I know that the drought and abiotic stresses, we have worked on biotic stresses too, are a major challenge, and how do we feed the 10 billion by 2050? One more so, next, giving credit to my authors. We need to understand that chickpea is certainly a drought-hardy crop grown in marginal lands. Nevertheless, it is quite prone to abiotic stresses, particularly at the seedling stage, at flowering stage, as well as at the pod filling stages. It is one of the largest source of protein to the greatly vegan population. Yesterday it was being said that about 39% of the Indian population is vegan. And of late, if you know, the vegetable protein industry grew from just $1 billion in 2018 to the current $120 billion industry. And the largest vegetable meat producing company has come nowhere but in UAE. So you can know that how vegetable meat or vegetable protein is going to play a major role in food. And with chickpea there we have achieved self-sufficiency. And it has the best amino acid profile that we can actually look into. Next please. Now if you look down while we were looking at resequencing of about 429 chickpea accessions, what we basically saw was when we started moving from wild species to that of the land races to that of the cultivated varieties, you see that a major chunk of genes which were basically non-selective or redundant genes which act as buffer remain constant. However, the genes both for crop improvement as well as for domestication, they greatly reduced. There was a large amount of gene chunks that have not been exploited from the wild species to land races, and so more so in the case of what we call as between the land races and the cultivated races. Next. Now, while we were looking at the abiotic stresses and looking at the growth parameters, this has opened up eyes. When you start looking at cereals, most of the cereals cessate their reproductive growth or vegetative growth and start their reproductive growth. So as a result of which, breeding for higher harvest index gave greater returns in case of green revolution in case of cereals. But when we start looking at legumes, be it pulses, be it soybean, the reproductive part is axillary, it's simultaneous, the pod first develops and then the seed starts developing inside. So you need to remember that it's not just the harvest index that improves yield in case of legumes. We have conclusively seen through our multi-location studies that it is the greater initial biomass that actually converts into greater yield. Next. So we started looking, are there any genes that were responsible? One of my students, Manu, was working on this, and then he checked out that there are different genes working just like yield in case of biotic stresses. Here particularly it was to drought, which were expressing. Earlier if you have seen that in case of USAR 362 and 1103, which expressed higher biomass, were actually working through abiotic abscisic acid synthase vitamin G, or in case of ICC 4958, USAR green 112, they were acting through dehydrin genes. Now when we started looking at this dehydrin gene, I'm just rushing through because I don't have so much of time down. It is one of the most conserved genes, but then there were very few what we call as Smith transitions when you look into the pool of those lines which were found to be universally informed as drought tolerant, whether it was Loxy from Ethiopia, whether it was ICC 495, they lined a land race from Kanpur, documented at Iktisad. Next one. And for the first time we started looking at other group of genes, and this lab has reported abscisic acid stress lightning gene, which was akin to most of the late embryogenesis 700 proteins, which were well characterized. Next one. And then this ERF family gene, which we started looking at, there were about 120 putative ERF family genes, and this putative transcription factor 116 not only encoded the protein family, which gave resistance under heterologous transformation to drought, but also for heat and also for cold. Next. So at the same time, my colleague and a close friend Varshney was working at Iktisat on actually tagging these root QTLs, and they developed a mapping population where we could identify QTLs that gave greater volume of root as well as root length. And moreover, in the LG4 where it was localized, we could get about 12 traits including harvest index, seed size, and the sort, and explained almost 58.2% variation. Next. So we started looking, because this was a large region, we thought that can we actually fine map this. Next one. And we could actually bring down this region to about 113 kilobase pairs, and we had about one more extra candidate gene that was coming with respect to number of branches per plant. Next. Now we started looking, can we validate these genes both in heterologous, which Ludwig has done and it has recently been published, and also in the 3000 accessions which we had sequenced. So we could actually identify and prioritize 3 candidate genes in this particular region which were actually providing drought tolerance. So the next idea was to mobilize this gene. See, the whole thing is you have the germplasm in the walls. You have identified either through the biotechnological processes or you have validated through physiological processes, but the utility or the end product makes its viability into a cost-benefit ratio. So we thought that we need to mobilize this, but then we need to mobilize in high-yielding varieties to show that drought-tolerant chickpea can be made more drought-tolerant. So we have taken a mega variety. A mega variety is a variety that has about 400 quintals of breeder seed production. So if you look at 400 quintals, by the time it reaches, it is almost 20 into 20, so 40 times, so it is almost 16,000 quintals. So that is the amount of acreage, because if you look at 100 kgs per hectare, that is the acreage. So we started mobilizing these QTLs from ICC 4958. We kept the genomic intact region by looking at the flag QTLs, and the NCTGR 21, 27 and CAN 23 are what we call as the gene-based markers, and we mobilized it. And the end product is keeping the background same as PUSA 372. We could develop a line which was similar to PUSA 372 phenotypically, but had better root traits even than the donor line. Next one. So what we have done is we started evaluating before it was entered into the national trials, both at field levels at 15 locations, in the Raynaud shelter and the DC scan facilities at Iktisat and Glasshouse at IRL. Yes, next one. And we could identify not only in case of PUSA 372, in case of JG11, ICC V10, a set of interrogation lines, the next one, which not only had greater drought tolerance, but incidentally yielded greater than the national checks. Next. So this was now pumped into the All India Coordinated Research Project trials, which was conducted at almost 8 locations, which were drought tolerant and the Raynaud shelters. And if you look at the moisture at the time of flowering, as well as moisture at the time of maturity, it is something between 15 to 20, and compared to the recurrent parent, it gave a whooping 11% more yield. Next. And then this has been dubbed as the first drought tolerant variety, which has been bred by genomic assisted breeding in case of anywhere in the world. More so that when we had severe drought 4 years back, and Gates was visiting the fields of Latur as well as those in the Vidarbha region, he found that nothing was growing but except that this chickpea was growing, that he has indicated to the Prime Minister that he would like to visit these fields at Ayar. Next. We went on to develop a few more varieties for other zones too. This is Northwest Plain Zone, which includes the desert regions of Rajasthan. Both these varieties have been developed. They are all in the seed chain. But what we are looking at, because Pusa 362 was also a mega variety drought problem, we were looking at also maintaining the greater biomass. Now what happens is you need to understand the variation between drought in central zone to that of the north zone. In case of central zone, the drought is gradual after the summer solstice or Sankranti, what we call as March 14, where the temperature slowly rise, so the root has still greater role to play. No doubt that in north also it plays, but what happens is we are having a cold. Somewhere around March 10th, you will see the temperatures from about 28 to 30 suddenly shoot up to 40. And here it is the above ground traits, and in this mapping population which we have recently published with Professor Jain from NIPD, we also identified QTLs for related water content and membrane stability index. And while breeding for this Pusa 362, what we have done is we have introgressed those QTLs. Therefore, if you look, the lower root system is like ICC 4958, from where we have introgressed the root donors, and the above system is akin to that of Pusa 362, which is drought tolerant, high yielding, and also has what we call as better QTLs for related water content and membrane stability index. Next one. Just in this January, we have released another super chickpea variety. We call it as Pusa JG16, which has higher yield compared to the donor as well as greater seed weight. Next. Now, that's what we say that our research had no boundaries, so we had gates in the field. But like morning, someone was saying, it's collaborations that play a major role. This was not a variety that was bred by Bhardwaj group alone. We had collaborations of Ikrisat, we had collaborations of the local partners, for example, in this case, both from Indore and case of Jabalpur. Next. Another particular stress that was playing a major role was wind, and we wanted to look in, because we didn't have wind resistance, and that was no problem in case of chickpea growing in the country, particularly in the central zone, which is the world's largest chickpea growing area in the world. So we started first looking at what are the toxin signatures and what type of genes play. Are there any QTL hotspots that have been identified? So working closely with NIPD and followed by University Spain, what we started looking is we started looking at what are various genes that are being upregulated in stress. I'm not going into detail. Next. Oh, I know you can see. So we have seen one crosstalk which is basically happening, that the heat shock 83 group of genes, which basically are the R genes, are also playing a major role, not only in case of heat stress in chickpea, which one of my students published in BNC in the earlier slides you have seen, but that particular gene also plays a major role in case of wind tolerance. Next. So further we have found out some miRNAs. One of my postdocs is now working on looking at gene silencing of this. Next. So then we started mobilizing these genes, which are in LG2 in case of chickpea, and we developed the Pusa chickpea manner, which we mobilized into 391 and grouping 153 printers this year we have for breeder seed production in the first year itself. Next. So you can see this is our grid set plot, very much in the field, and madam, if you can get the students, certainly we will be showing them. These are whole strips of 200 meters. This line has vanished. Pusa 362, which is a susceptible line. This is the donor line, and this is the manor. To the whole 200 meters, you don't see, and this is the new line that we have developed in the background of JG11. You can see where JG11 has succumbed, this line is totally resistant. Next. We have developed a large number of varieties across India. As I say, these are for drought, for example, Pusa GB16 with 2.2 tons under drought stress conditions, 1.9 in the northwest plain zones in the hot stress, drought stress conditions of Rajasthan. Pusa Chickpea 10216, the first marker resistant variety, 3.5 tons, and Nano with a whooping 3.94 tons. It is giving a big run to beat in case of central India. Next, please. Now, what I mean to say is, when you get on to the robust genes that have been identified, these genes have been well documented and validated, not for publication, but well validated, and then they are introduced, they work across the globe. Be it in case of India, my friend Asnan Parake, who has worked in case of gelatin in Ethiopia, or in case of Kenya, we are having, so these lines have been released as traditional, as high-yielding varieties, more so that these lines have been continuously being dedicated. This was last year's slide, before last year. The two of these lines have been dedicated to the nation, the Honorable Prime Minister. Next, please. So in brief, if I conclude, just taking 16 minutes down, that the next generation genomics are here to stay and here to stay. But the most important thing in case of any crop improvement program is the basic genetic resources which I was speaking, the 3 Gs, that you need to have a good genetic resources, you need to go ahead with developing the good genomic resources that are there, the validated genomic resources, and the genomic assisted breeding technologies and the breeding methodologies, for which high throughput phenotyping is required. While we are starting to move into the genomic assisted breeding 2.0, where we have started a genomic selection program with Gates Foundation as well as with Indo-Canadian Pulse Genomics Initiative working with Saskatchewan, what we are now looking at is going at genomic selection, where we are trying to basically improve the breeding value. If you start looking at the last 10 decades, in rice we had just 2 cages per year increase. From 1950, whooping 200%, but hardly 0.1 increase. In case of chickpea, it is 1.5 kg per year. It's very, very low when you look at nation as such. This can now, we need to, like Green Revolution has done a quantum jump by bringing out genes that were playing a role, like RHT, the barking gene in case of wheat, or in case of DGO, the gene in case of rice. So you need to start looking at ways now how to keep an increase. And this now, because since you are exploited that are the single major gene or pleiotropic genes, you need to look at bringing in the whole genomic pool, which we need to improve. That is one. Second, looking at genomic improgressions and capturing on how we are able to use those whole data and move them to haplotype breeding. So this is what we are working in the next few years. Next, please. I'm thankful to all of my co-authors. And next, And most importantly, this is certainly not a work of mine, but of my students and my research fellows that have done. Thank you, Dr. Vani and Suman Madam, for giving me an opportunity. Thank you.